Sequencing and Introduction to HALE. This is your third video, Next Generation Sequencing Technology, for the 2021 virtual workshop on StatGen methods for human complex traits, hosted by the Institute for Behavioral Genetics, University of Colorado Boulder. My name is Kumar Virpan, and this module would be TA'd by Tim Perturba, Caroline Diaz, Dan Horrigan, and John Compitello. This video would be the first thing to understand the overview of next generation DNA sequencing methods. If you listened to the previous video, you know why I put it in air quotes, and in this case, actual quotes. The sub-learning objective, the first one, would be to appreciate the rationale in moving from Sanger sequencing and other next-gen sequencing methods to capture the concepts of sequencing by synthesis, to understand that there are a variety of applications for sequencing. And so I'd like to just bring us back into understanding that when you run a GWAS, you can use both genotyping chips and whole genome sequencing. The difference is, if you were to think of maybe like a textbook, in the genotyping methods, you would only be capturing maybe like, I don't know, a paragraph a chapter, or a few paragraphs a chapter. For whole genome sequencing, or WGS, you'll be capturing the entire book for the most part, and, well, every part of every chapter. So in this case, the chapters would be all the chromosomes as represented here in a Manhattan plot, and an LD plot here, right here. So to refresh our memory a little, on the left would be the sequencing Sanger method. On the right would be what we're going to go through in this video. So on the left, when we do run a sequencing by Sanger method, if I were to draw your attention to the third line here, where we have the multicolored sequences with the dideoxy nucleotides ending on each of them, and then reading it through a capillary, and finally looking at the chromatogram or the spectrogram for you as the reader or the user to interpret. So we would, for both methods, Sanger sequencing and sequencing by synthesis, you would fragment your DNA into pieces. The difference is here you would then amplify using PCR or any kind of in vivo cloning method. In this case, you would use an adapter ligation in order to ligate at well, kind of semi-random. You have like set aside sequences in your tube of adapter ligators that you put into your reaction tube to ligate or attach onto these DNA fragments, and then forming polonies, which I'll go through what they are, and finally what the sequencer will take, which is pictures that will tell us what is base number 1, base number 2, and base number 3 for each of the polony. All of these words, polonies, and sequencing by synthesis, hopefully will be very clear to you by the end of this video. So sequencing by synthesis, or as short here is SBS, I took this from, actually to be really honest, I can't remember the source of this, which I apologize. So as we mentioned previously, what happens is you take your whole genomic DNA, you will fragment it up into pieces. You will then attach ligators onto it, which is kind of like primers if you think about it. And then this would just like specifically take that these regions are what you want. So these ligators or adapters is what would be really important when it comes to like maybe exome sequencing, where you're only capturing just the exome. For the genome, you will capture, well, the genome. Alright, so upon capturing this, you would then incubate it a few times, a few experimental steps in between, and then you'll put it on a glass slide chip that each chip has about 8 capillary-like lanes. They're really, really tiny lanes that you would then elute your reaction into. In the early 2010s, we would actually run one sample per lane just to have as high density sequence as we can. Now we do something known as multiplexing, where one capillary lane you would put multiple samples in. So now that we have the samples in, the DNA will attach onto this glass chip. And this glass chip, as here it says, one strand of DNA is firmly anchored onto the end of the glass chip. This is within the capillary itself. So then you would incubate it with a primer, and this primer is just complementary to what you have captured earlier using the adapter ligands, and Upon incubating it, you wash away the primers and you'll start your reaction. So I did say there is a rationale to understanding what PCRs are, which in this case would be to recapitulate what primers are used for and how they work. Secondly, the Sanger sequencing method came about after the PCR method, and the dideoxy terminal is what you can see kind of here. So what happens here is you have this reaction within the capillary itself. You would include artificial nucleotides here that would be having the A, T, G's, and C's, that each of them would have a specific dye color. So you would incubate it, only attach one where this blocker here would block any other nucleotides to attach, regardless of all the other artificial nucleotides which are present. So at this point of the reaction, you would have the primer sequence and just the first nucleotide with the blocker on it and the dye. So in the next step, when a laser light is moved onto the capillary itself, what this laser light does is it would cleave away the blocker and it would cleave away the dye. So this in itself is a really interesting and most important reaction here, because at this reaction, two things occur. One, when the dye is detached, the sequencer will capture a picture like what we saw previously. What is base 1? What is base 2? What is base 3? And that would be the picture for base 1. And so in this case, base 1 would be red. And then the process begins again by changing the temperature to allow annealing of the next nucleotide. I'd like to remind everyone that this occurs in hundreds of thousands of reactions on a single capillary. So it's not just this one, and it's hundreds of thousands of multiple different strands that could look either the same or different. So that's why every single time each nucleotide is attached, a picture is taken, and this picture is saved, which will then be parsed out. So this is how a colony looks like, and this is what I was mentioning with the hundreds of thousands or sometimes clusters of molecules where each of them will be known as a colony. So this colony is where you would have formation of these loops, and then you would repeat the reaction and repeat the reaction, which is why you have multiples of a single strand of your already cleaved and amplified sequence. So you would have about 32 million clusters per experiment, and these clusters would be your polys. Upon the end, you would have gigabytes of sequence data. Why? Because these random clusters here, if you recall, every time you start a reaction and a nucleotide inserts in, a picture is taken by the sequencer. Pictures are high, well, they're not high intensity, I was going to say high storage needs, has high storage needs, which is why each picture of each reaction, and this goes on for about 99 to 101 reactions in total, just because the nature of each of these segments would be 99 to 101 length. And that's actually one of the important things when you think about like fragment lengths, when you do some of the informatics downstream. This would just be a different way of expressing what I was mentioning earlier, where you would attach, form the bridge then cycles through amplification, denatures the product. The end results would be polonies of the same PCR reaction. You would then do whatever we went through in the beginning, where I was mentioning each nucleotide going in one at a time, and you would image each base at each polony. And after each additional sequence, you'd have cycle 1, cycle 2, cycle 3, cycle 4, cycle 5, cycle 6, until cycle 101, where each polony you would have one whole sequence formed out from your entire, from your sequencing read. So these are some of the pictures that you could observe, and so this would be an example of one of the polonies. This would be an example of another polony that you would have the reads come off of the sequencer. So the sequencer looks really like this. It's a huge machine, but now they have tabletop sizes too, almost the size of your monitors. But of course, they're wider and a little more dense, right? So in the sequencer itself, this would be where the laser light protrudes onto or shines onto the capillary chip, and this would be how the capillary chip looks like. Nothing truly fascinating, but there are 8 lanes usually in a capillary chip that you can multiplex depending on your experiment, even up to 50 samples per lane. And this is something that I would highly recommend when you talk about sequencing. Talk to your genomics platform people and ask about what is the recommended amount per lane. They would usually figure it out for you. And usually the more you have per lane, the cheaper it is. Yeah, so that would be some of the reactions. So to give you like how old I could possibly be, I remember when we ran per lane, it's about $4,000 into 2013 if it's one per lane, and you just divide $4,000 with the number of sequences you have per lane. To be really honest, I've not done this experiment in a very long time, so I don't really know how much is it per lane anymore. It could be 2,000, 4,000, but I do know that the cost per genome used for research purposes can be down to about $1,000 or $800 per whole genome, which is far less money to spend as opposed to 4,000 in 2013. So to appreciate this more, there's multiple different omics, if I would put it, as applications. The top line here shows the central dogma of molecular biology. DNA transcribes into RNA, RNA translates into protein, and the protein would devolve into the phenotype. So when we think about omics and how next-generation sequencing can help with this, DNA would be the genomics, and the modification of DNA would be the epigenomics. Transcriptomics would be the RNA. Proteomics would be protein, which is a very different technology compared to next-generation sequencing. And metabolomics, again, very different compared to the next-generation sequencing that we have gone through, that I would rather call high-throughput sequencing or sequencing by synthesis. All of this goes through a hefty bioinformatics pipeline that, to be really honest, I do not have the true expertise to talk about, but I will introduce some of the concepts to you in our future, I think it's our next video, actually. So we'll be focusing a lot on the DNA and less on else. So this slide just shows you how some of the applications for genome sequencing and all of the different applications taken from Jason Dury's 2008 paper that I did highly recommend to read if you'd like to just appreciate the historical nuances of next generation sequencing. So the learning outcomes for this, well, relatively short video here would be you understand the overview of what NGS or high-throughput sequencing methods are. You appreciate the rationale in moving from Sanger sequencing to other NGS, in particular the high-throughput sequencing for sequencing by synthesis, because, 500 base pairs in Sanger sequencing is nothing compared to millions of base pairs you could get from NGS. Subsequently, you can capture the concepts of sequencing by synthesis. I had 2 different ways of describing it. First, by going through just the method itself of each nucleotide going in, laser going on, fluorescent coming out, and each picture is taken. And subsequently, I also talked to you about how the polonies are formed and how these polonies lead back to the concept of the lasers being lit onto each of these reactions. You also understand that there are a wide variety of applications for sequencing, which I have gone through rather glacially, just because those are more for like your additional reading, not truly important for this module or even for the end goal of the IBG. Thank you. My name is Kumar Veerapan. If you have any questions, feel free to ask me a question on Slack or send me an email at veerapan or at broadinstitute.org. Thank you.